Hello, student. This is chapter 6, Anatomy of Flowering Plant, and this is video number 4. In the previous video, we have talked about the anatomy of diploid and monopod plants, the root, stems, and leaf. And in this video, we'll be talking about, I'll be explaining you about the secondary growth. Now, we all know that the meristematic layer that is responsible for cutting out the vascular tissue, that is the xylem and the phloem, and that is known as the vascular cambium. And in the young stem, it is present in patches as single layer between the xylem and the phloem, which later forms a complete ring. Now, how is this complete cambium ring is being formed? You see this cambium ring in the diploid plant, not in the monopod. In the diploid stem, the cells of the cambium present between the primary xylem and the primary phloem is known as the intrafascicular cambium. Now, the medullary veins adjoining this intrafascicular cambium becomes meristematic and forms interfascicular cambium, and hence a continuous ring is being formed. You can see in this diagram too. Now, what is the activity of this cambium ring? The cambial ring becomes active, and as it's become as a meristematic, it becomes active and begins to cut off new cells, both towards the inner side as well as the outer side. The cells cut off towards the pit, that is the inner side, forms the secondary xylem, and the cells which are cut towards the periphery, that forms the secondary phloem. Now, children, the amount of secondary phloem produced is much more than the the amount of secondary xylem produced is much more than the secondary phloem. And so soon it forms a compact mass. At some places, the cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radial direction, and this is known as the secondary medullary rings. Now, you must have heard about springwood and autumnwood. In the spring season, the cambium activity is very active. The activity is more, and it produces large number of xylem elements having vessels with wider cavities. The wood formed during this season is called the spring wood or the early wood, and you can see over here also that it is of lighter color. In the winter, the cambium activity is less. Cambium is less active and it forms fewer xylem elements. That is, the vessels are narrow and the wood is called the autumn wood or the late wood. So, every spring season, early wood, every winter season, late wood, together both forms one annual ring. So, in 1 year, one early wood, 1 year. So if you want to know the age of the plant, you count the annual rings, you will come to know the age of the plant. Now, let us talk about heartwood and sapwood. The region which comprises of dead elements with highly lignified wood, that is called the heartwood. And the peripheral region of the secondary xylem is lighter and in color and is known as the sapwood. You can even see it in this diagram. The inner part, which is dark, is the heartwood, and the outer part, which is lighter, is the sapwood. Now let us talk about fork cambium. What is fork cambium? As the stem continues to increase in girth, because you have seen the vascular cambium is active, and it cuts out xylem and phloem. The outer part, so the more of xylem is formed on the inner side, so it puts a pressure on the outer side, the outer cell, and the outer cortical and the epidermal layers break and need to be replaced by another meristematic tissue, which is known as fork cambium or phalange. Now, the outer cell differentiates into fork or phloem, while the inner cell differentiates into secondary cortex or the phalange. Children, this fork cambium or phalange also starts cutting out cells on inner side as well as outer side. So the outer cells, they form the phloem, and the inner side, it gets differentiated into phloem. Together, the phloem and the phloem is known as periderm. Now, outside the periderm, there is bark. Bark, it is the tissue exterior to the vascular cambium, and therefore it includes secondary phloem, that is periderm and secondary phloem. Now, let's see the bark. You can see over here the bark also. Here, you can see here the bark. It's the living phloem. The bark is formed early in the season and is formed early or soft bark, and towards the end of the season, it is the hard bark. And you can see here the periderm, just after the periderm, the bark is there. The parenchymatous cells soon rupture the epidermis, forming a lens-shaped opening called lenticels. Now, the epidermis, it has an opening that is known as lenticels, and from there, exchange of gases may take. Like in leaves, we have stomata. In plants, the opening is the lenticels. Now, let's see the secondary growth in roots. That is the diploid root. That also shows the secondary growth, just like the secondary growth in diploid stem. The vascular cambium originates from the tissue located just below the phloem membrane, a portion of pericycle tissue above the protoxylem, forming a complete and continuous wavy ring, which later becomes circular. In the diploid root, the vascular cambium is completely secondary in root, and the development is exactly similar as that of the secondary, as that of the diploid stem. Thank you. With this, we finish the chapter.